Good morning, class. Hope you guys had a good week so far. We're going to get started right now. So the purpose of a tutorial is once again to take up the questions from the study guide. And the idea is that I assume you have watched the recordings already. And so I'm not going to go into a lot of details regarding some of the concepts. I would just kind of condense it and give you a summary of the topics. So for the more in-depth explanation, you would have to go back to the lecture recordings for that. So this is the scientific method study guide. If you have not download a copy, you can get it from the link in the chat box that I just posted. And so let's get started. Here, first question. And again, right, like if you have any questions during the tutorial, feel free to stop me. Just unmute and ask the question or type your question in the chat box. So here we go. List 7 characteristics of living things. Okay. Anybody want to give me some examples of characteristics of living things? You can just type them in the chat box. Okay, so very good. There is the first one. They exhibit order. Okay, so that means, for example, there is going to be some symmetry. Okay, there's going to be some complexity. Okay, so exhibit order. Another one is I see in the chat box, response to stimulus, homeostasis, very good. These are all very good. So we will do ability to maintain homeostasis. Okay, homeostasis means what? Can someone tell me the meaning of the word homeostasis? What is the meaning of homeostasis? Anybody? Okay, so homeostasis is an internal balance, right? An internal balance. So despite the fact that the surrounding changes all the time, temperature fluctuates, for example, your body might have increase in sugar level or decrease in sugar level, depending if you're eating or fasting, but you are able to maintain homeostasis. You're able to maintain an internal balance. Now, in order to do that, in order to maintain homeostasis, organisms can either do it through what we call physiological means. Okay, or they can do it through behavioral means. Okay, so physiological means that your body will adjust itself. The example I gave in lecture is that if it's cold outside, you will start shivering. Okay, so shivering is a physiological response to a drop in temperature. The shivering will heat up your body and make you warmer. The behavioral response to that would be putting on a jacket. Okay. Another characteristics, I'm just looking at what you guys type, all are excellent points. Growth and development. Right? Okay. We look very different now than when we were a baby, right? We have been growing all this time and developing, and the growth does not necessarily stop, right? It keeps on going. Another one is requirement of energy. Require energy. Okay, so we do this through what we call cellular respiration. Right, to produce energy. Another one is reproduce. Okay, organisms need to reproduce, to propagate the species. Another one is respond to stimuli. Okay, so stimuli are basically changes in the environment. Okay, and these changes is going to cause your body to respond in a particular way that would help it maintain homeostasis. Okay, so not just humans, right? Like plants, they do these as well, right? They're able to respond to changes. They grow towards the light, for example. That's an example of responding to stimuli. And last but not least, let's see if anybody mentioned this so far. Yep, Carl mentioned it earlier. Adapt and evolve. Very good. Okay, so remember, for evolution, it's important to know that individuals do not evolve. Okay, population does. Okay, so just to briefly give you an illustration to help you understand this or remember this. Let's say there is an island, and on this island, there are these little organisms, okay? So these organisms comes in two forms, okay? There are the ones that are naked, they don't have furs on them, and then there are ones that are furry, okay? So there are two groups of populations here on the island, okay? The furry guys and the naked ones, right? And so what happens is, let's say there is some kind of environmental change, sudden environmental change, cause the temperature to drop. Okay, so what happens is the furry ones will be okay because the fur keeps them warm, but then the naked ones, they are going to probably suffer from the cold temperature, so they might die off. Okay, so the ones that get to live on will be able to mate and have more offsprings than the ones that are not surviving or barely surviving, right? So over the course of several generations, okay, several generations later, you're going to end up with a population that are mostly going to have these furry guys, okay? Just copy and paste a bunch of them. There we go, okay? And does that mean you're not going to have these naked ones? No, you might still have one or two, okay? But they are not going to be the majority of the population, okay? So because of a change in the environment, okay, the entire population will shift towards traits that will allow them to survive better, okay? This is evolution, right? Evolution does not mean the naked one will all of a sudden start to grow fur. Okay, they genetically do not have the fur. They're not going to grow the fur because the temperature drops, right? Okay, but the entire population will shift towards this favorable trait in this particular condition. Now, suppose if the temperature increase in another time, then the furry ones are not going to be doing too well, right? And so they start to die off and not have as many kids. And then the population might shift towards the other trait that is more beneficial. Okay, so that's basically what evolution means. Does anybody have any question? All right. Then in the lecture, we talk about this concept of induction and deduction. So let's just write down the definition for each one of these first, and then we will give some specific example. So for induction, the definition is basically we derive is just a fancy word of saying coming up with. Okay, so we come up with a general statement based on many specific observations. Okay. This basically goes from specific to generalization. Okay. If you have to represent this on a diagram, it's like this. Okay, so we make one observation, we make another observation, and another observation, and another observation, and then we keep on doing it. And then we have a bunch of observations here, or a bunch of examples here, and we take all these observations that we made, and then we are going to make a general statement. Okay, that is essentially what induction means. So an example of this would be, let's just say, statement number 1, birds are made up of cells. Fish are made up of cells. Okay, and then sharks. I guess sharks is kind of like fish. Bees are made up of cells. Okay, so these are specific examples. So what's the general statement? General statement. Like obviously you're gonna need more examples, right? But I hope you get the point. So the general statement would be all animals are made up of cells. Does anybody have another example they would like to share with the class? Another example of induction? Okay, so here's another one that may be more relatable at the current situation. Okay, let's say Janice went to Peter's birthday party on the weekend, and she has COVID now. Okay, Mary also went to the same party, and then she had COVID. Nicholas went to the same party, and then he has COVID. Right. So we see that all these people went to that party, and then they have COVID. Then the general statement I can make, right? The general conclusion I can make is that, oh, someone must have had COVID at the party, and then they are spreading it to everybody. Right. So from specific examples to generalization, to a broad statement. That's basically what induction is. Now, deduction is the opposite. Deduction goes from a generalized statement to a specific example. Okay, so general to specific. Okay, so using the general statement, we can make a prediction. Okay, so using the same example that I have here with the animal things. So birds are made of cells, fish are made of cells, and then we made a general statement that all animals are made up of cells. So the deduction from that would be, well, dog is an animal. Therefore, they are made up of cells. Right? So from this general statement, right, we are going to go and make a prediction for a specific case. That is deduction. Okay. So back to the the COVID and the party example, right? How would I go from that general statement and make a prediction, right? So the induction was saying all these people went to the party and then they had COVID, so therefore someone must be spreading it at the party, right? That's the induction part, right? The deduction part would be, oh, I also went to the party, right? Therefore, I should get tested to see if I also have COVID. Does that make sense? Any follow-up?
Yes, I'm understanding.
Okay, thank you.
You're welcome.
Anyone have any follow-up questions? All right, well, let me give you a question to see what you think. So here is a question for you to try. So I'm gonna launch the poll and I'm gonna give you a minute or two. And please take a look and let me know. So this would be an example of a question you might get on quiz or something. Okay, just half more minute and then we're gonna take this up. Please choose something, even though if you're not sure. All right.
Can you send the poll, please? I couldn't see the poll.
I launched the poll already. You should see a poll button, but sometimes it doesn't work for some people on certain devices. In that case, it's okay. You can just, try to do the question in your notebook, and then we can take it up. Thank you. All right, let's end the poll. Most of you have answered. Here is the poll result. Most of you choose B or C. So let's take this up, right? So the question asks for an example of deductive reasoning. So you should know deductive means going from general to specific, right? Okay, so let's take a look at the first one. All viruses requires a host to replicate. COVID-19 is a virus. It must require a host to replicate. Okay, so is that deductive reasoning? Is that going from general statement to specific statement? Maybe. Okay, let's take a look at the next one. This is COVID-19, Ebola, influenza are viruses that requires a host to replicate. Therefore, all virus requires a host to replicate. So this is example 1, example 2, right? And then you make a generalization. So this is actually induction, right? Going from many examples to the broad example. So this is not an example of deductive reasoning. Do you guys understand why this is not? Many of you chose it, but have I just convinced you that it is actually inductive, this one. Okay, remember, inductive is from many, many, many examples to a broad statement. Okay, so that's not. Next one. All virus requires a host to replicate. COVID-19 requires a host to replicate. Therefore, COVID-19 is a virus. Okay, so maybe, right? This one is also maybe. We can come back to it in a second. Let's take a look at D. COVID-19 is a virus. It requires a host to replicate. Ebola is also a virus. Therefore, it also requires a host to replicate. So this is, again, not the answer because you are just stating one example and then you're just stating another example. Okay, you're not really making a generalization. Okay, or you're not going from a general statement to a specific statement. Okay, so that is not the answer. So here is either A or C. Now, many people chose C, right? Let's take a look at it again. All virus requires a host to replicate. COVID-19 requires a host to replicate. Therefore, COVID-19 is a virus. Now, at first, it seems like this makes sense, but let's change it to, you know, like a different wordings, and you will see what the problem of this statement is. Okay, so here, instead of saying all virus, we will say all birds have wings. Okay, so instead of saying require a host to replicate, we'll say have wings. All birds have wings. Okay, and then he said COVID-19 requires a host to replicate. So instead of saying COVID-19, I'll say bees, right, requires a host to replicate. We'll say bees have wings. So like going in the same format as this statement, then what's my conclusion? My conclusion is that therefore, COVID-19, or in our alternate example, bees is what? What do I fill in the blank here? Do you guys follow the logic? Yeah, bee is bird, right? And you can see with the bees and the bird example, what is wrong with this sentence structure, right? Okay, so this is not correct, right? So that means deductive reasoning. First one, okay, broad statement, all viruses requires a host, and then you apply it to specific example. All right, so only one person got it right. That's okay. That's okay. If you see something similar on the test, hopefully you will get it right. But does anybody want to ask any question about this? Any clarification?
Professor, I missed the first class because I didn't know how to use the timetable. I was trying to log into my chemistry class, and there I had a biology class, not a chemistry class.
Right. Now, there's no first class, right? On the Tuesday, you're supposed to watch the recordings. So during the break, I'll post a link to the orientation so that you will know how you can watch that and you can see how the course is run. Okay, so the first class is just watch the lecture recording on your own. Okay, so technically you did not miss anything. Emma asked replicate, meaning to make more copies of themselves. Okay, so like you have one virus and then they replicate, it means they make more. Okay, make more. That's what replicate means. All right, let's keep going. List the steps in the scientific process. Okay, so first step is to make observations. Next is ask questions. Okay, then you are going to try to answer your question by stating a hypothesis. Yeah, hypothesis. Okay, so hypothesis is a tentative answer to the question. Okay, it might not necessarily be the correct answer, but is a proposed answer. And in order for a hypothesis to be valid, it must satisfy two conditions. What are the two conditions, two criteria that must be satisfied for it to be a
Yes, and
valid?
law.
I'm sorry?
It
Once
should
again?
gain the universal law, or no. It should be positive or negative.
Okay. Anyone else? Okay, Patricia said, yeah, has to be testable. That means you can run an experiment to test it, and it must be potentially wrong, so falsifiable. Falsifiable. Okay, potentially wrong. Okay. After you make the hypothesis, you are going to make a prediction. Okay, if my hypothesis is correct, then I would expect these outcomes. If my hypothesis is not correct, then I would expect, that. It's also important to know that the hypothesis is not a question. Okay, when you do your lab and I ask you to write a hypothesis, it should be a statement. It should not be a question. After making the prediction, you would then go ahead and do the experiment. And from the data, you will do a conclusion. That's the steps in the scientific process. Give an example of a non-testable hypothesis. Anybody have an example of a non-testable hypothesis? Yeah, any opinions are non-testable, right? So for example, biology is better than math. Okay, that's a statement, right? You cannot test that. Religious belief, myth, that's right. All those things you cannot test. Someone in my other class says Beyonce is the best. Okay, that's non-testable. Okay, it's an opinion. Give an example of a non-falsifiable hypothesis. Okay. Anybody want to try that? Non-falsifiable hypothesis. Plants receive more sunlight, grow taller. That's falsifiable. If I put the one plant in the sunlight and then one plant not in sunlight, and then it turns out that they actually grow shorter in the presence of sunlight, then that's falsifiable, right? What about hair number? Okay, so non-falsifiable example would be like this. Everything that happens is as a result of divine intervention. Okay, there is no way to prove that wrong. Okay, that statement is an example of a non-falsifiable hypothesis. Okay, here's another one that that my parents liked to tell me when I was growing up. If you work hard enough, then you will get A+ in whatever subject, right? Growing up, the subject was English for me. Okay, so that's non-falsifiable, right? Because if I don't get A, then they can always say, well, that's because you didn't work hard enough. Okay, and then if I work harder and I still did not get A, well, that's because you didn't work hard enough, right? Okay, so that's an example of a non-falsifiable statement. Natalia asks, how is this non-falsifiable? Okay, so you cannot prove it wrong, right? Because there is no way to prove whether something is a divine intervention or not a divine intervention, right? Okay, because that's something that you believe, or like the person believes, is personal belief, right? So we cannot prove someone's belief to be wrong, right? Okay, if they think, okay, this is happening to the world is having COVID right now, it's having a pandemic right now because it was meant to be from the divine intervention. There's no way to prove that to be wrong, right? Okay, it's just a statement, right? That the person believes. So as such, it's non-falsifiable. Okay. Whereas if I say that sunlight is causing the plant to grow taller, then that could be falsifiable, because if I remove the sunlight and then it stops growing, then I showed it, right? Okay. So there's no way to prove it wrong because there's no way to prove it right. Yeah, in a sense, right? Okay, if you cannot have hard evidence to either accept or reject it, then it's potentially non-falsifiable. Okay, does that answer your question? Okay, great. Moving on, give 2 examples of primary sources and 2 examples of secondary sources. Example of primary sources would be peer review journal, right? Peer-reviewed journals. So these are papers that are published by scientists, and before they publish it, other scientists, other experts in the field would scrutinize the data, look at the paper before they give it the go-ahead to release it. And these are primary sources because the author, the scientist who conducted the experiments, are the one who writes the paper. Ryan has another question. Could biology is better than math or Beyonce is the best also be examples of non-falsifiable? Yeah, that is also correct. Okay, you can test it, and usually the two kind of go hand in hand. Okay, very good. Okay, what's another example of primary sources? Anyone? Help me out here, guys. Yeah, could be survey results that are published by the people who are actually conducting the survey. Very good. Here, I'll put it here. Survey results. First hand, right? Survey results. Another one would be like reporter. Reporter doing a live broadcast at the scene of something. Okay, there is a car accident and the reporter is describing the scene to you live at that scene, and that's primary source. Okay. Secondary sources would be examples like textbooks. Okay, textbook usually takes a collective a body of information that are taken from various sources and then they put it together, right? So the person who wrote the textbook, usually speaking, is not the person who discovered the stuff. So if you read your bio textbook, it tells you, oh, you know, living things, they exhibit order, they maintain homeostasis. Like the person who wrote that did not discover that, right? Like it was discovered probably a long time ago by someone else. Okay, so that's a secondary source. Okay, newspaper. All right, it's not always primary source either. Okay, so if I am quoting someone, okay, if I interviewed a person and then now I'm just writing an article about it, then that becomes secondary sources. Okay, so Karina, the main difference between primary and secondary is this. Primary has to be the person who's writing the material has to be the person who experienced whatever they're writing firsthand. So if I did an experiment and then I write about the result of the experiment, that's a primary source. Okay. if you read about my experiment, if I tell you about my experiment, you interview me and ask me about my experiment, and then you write an article about it, then the article is going to be a secondary source because it did not come from me, right? It come from, you know, a secondhand source, basically. Make sense? Or you have a follow-up question? Okay, great. So obviously, if you are doing research, right, primary sources are important, right? Okay, so a lot of the false information that you find online is actually secondary sources, right? People pick and choose what they want, and then they twist the meaning to it. Okay, and then they write something about it. For example, I'll give you one common example. I read on the a newspaper, and then there is an article that says, polio vaccines killed 20 children because of improper transport condition. Okay, so that happened. The vaccine got maybe contaminated during the transport process, and then it caused harm in the end. Okay, but someone else might take that, and then they just twist the meaning. They will say, oh, polio vaccine kills children. Okay, so that's taken out of context, right? That's a secondary source, right? Because they took what they read, and then they manipulated the meaning of it, and then they post it, right? Okay, so primary source are more trustworthy. Secondary sources, you know, if it's a textbook, right, it should be okay. But then there are many secondary sources that are biased as a result of of what I just told you. Okay, so even newspapers, they could be biased, right? Like there are newspapers that have different political affiliation. For example, some are more liberal, some are more conservative, and then the views that they present in it might also be biased. Okay. Number 6, I did this already in the lecture recordings, but just to quickly go through these things, independent versus dependent variable. Okay, does anybody want to tell me what independent variable means? Okay, feel free to unmute if you are comfortable talking. If not, typing is also fine. What is independent variable?
The one which don't needs to connect with another one in order to survive, or
okay, good try. Not quite what I was looking for though. Anyone else? Independent variable?
Independent variable you can control or change.
That's right. Okay, so the independent variable is the variable. First of all, a variable is it's something variable is basic in an experiment. It's just a parameter. It's for example, like the height of a plant, right? The temperature, right? All these things are variables. The amount of water, for example. Okay, so the independent variable is the variable that is controlled or changed by the person doing the experiment. Okay. Whereas the dependent variable is the one that is not controlled by the person who's doing the experiment, but rather this is the variable that is being assessed. Okay, this is the one that is being observed. This is the one that is being measured. So in general, in an experiment, the purpose is to change the independent variable and then see if that has any effect on the dependent variable. Okay, so again, slowly, the purpose of an experiment is to determine if changes in the independent variable will have any effect on the dependent variable. Okay, so the way to remember it is, independent. There is in here, right? So independent variable is the one that you are in control of, the one that you are deliberately changing. Okay. Now let's just take a look at the first one. Again, I did this in the recordings already, but I'll do the first one here just as an example. Students watched a cartoon either alone or with others, and then rate how funny they found the cartoon to be. Okay, so the independent variable in this case would be watching the cartoon alone, or with others. Okay, so you can control, right? Okay, like you go watch it yourself. Okay, you guys go watch it together. And then the dependent variable is how funny was the cartoon. Okay, so does watching it with others make it more funny, right? Or does watching it alone make it more funny, right? So by changing the independent variable, we want to know if it has an effect of the dependent variable. Questions, anyone? Thank you, Karina. Anyone else? Going once, twice. All right. Okay, let's do another poll. This one should be simple. Go ahead. Okay, just a couple more seconds. All right. Tentative answer. That is hypothesis. Okay, most of you got it right. Very good. Okay, how about this one? Okay, this one is better than the last one in terms of people getting it right. Experiment, that's correct. Okay, let's move on then. So this one is about Francesco Redi and Louis Pasteur experiment on the theory of spontaneous generation. Okay, so spontaneous generation. What is spontaneous generation? So let's write a definition explaining the spontaneous generation hypothesis. So basically, the theory states that living things came from non-living things. Okay, so in the past, people were saying that all the living organisms must have spawned from non-living things. Okay, because people see, fishes rain from the sky, and they're like, oh, sky, non-living, fishes are living. Therefore, living things came from non-living things. Okay, but now we know that's a natural phenomena. Okay, like maybe a tornado swept through the lake nearby and then send all these fish raining down inland. If you do a Google search, you will see that it happens quite often. Another example was that if you leave a piece of meat outside and come back a couple of days later, you see maggots crawling all over it, right? And flies. So maggots, being the living thing, came from the piece of meat, which was non-living. Okay. So Rady, right? Rady is the guy who's going to be doing the experiment to see whether spontaneous generation was true. So the observation that he made his hypothesis on was basically what I just described to you. When a piece of meat is left outside, flies and maggots will appear on the meat. Okay. So he did an experiment to see, if this is actually going to support the theory of spontaneous generation. He has 3 groups. The first group, if you remember from the lecture, is that he has a jar. Okay, and this is an open jar. It's not covered, and he's gonna put a piece of meat in there. Piece of meat. Okay. And in group number 2, he has the same jar, but he will put a mesh netting on top. And again, we have a piece of meat inside. So this is jar with, netting. And then he has a third group. He has a third group where it is going to be covered and sealed. Okay. And of course, we still have the meats in there. There we go. Sealed jar. Okay, so that's the 3 groups right here. Now I want to ask, what is the independent variable in this case? For this setup, what's the independent variable and what's the dependent variable? Anybody? What's the dependent variable?
I think the independent variables are the types of jars used.
Excellent. The types of jar. Type of jar used. Right. He controlled whether to have an open jar, netting jar, or sealed jar. What about the dependent variable?
I think the result of the experiment.
The result of the experiment, okay. So the result of the experiment will be the measurement or observation of the dependent variable. But what exactly is that dependent variable? A lot of people saying the meat, but it's the same piece of meat. Okay, if I put a piece of beef here, it's gonna be the same type of beef in all of them. Emma said flies. Can we be, yeah, so like the presence, right? Presence of flies and maggots, right? Okay, so that is the dependent variable. By changing the type of jars, we're going to see if that will affect whether flies and maggots showed up. Okay, if you have not read or watched the Radius experiment yet, you should do that. Okay. Remember the video resource link that I showed you last time? There is an animation in there that you should go check out. Okay. What were the results of Rady's experiment? Okay, so we have 3 groups. Let's just summarize the results. Okay, well, group number 1. Okay, any maggots and flies in group number 1? Well, as you expect, there will be both flies and maggots. Okay, so you're gonna have flies all over the place. Okay, and then you have these disgusting maggots growing on them. You know, just a quick story. Every summer, I don't know why, but my garbage can will be infested with like maggots and flies, and it's so gross, right? Like I try to take the garbage out, and then I see all these maggots growing on it, and then I can't deal with it. It gives me goosebumps. My wife is totally not bothered by it, so I would call her, and then she would come and clean it every time. Excellent help. But the maggots are very, very gross. Group number 2. Any flies and maggots on group number 2?
Yeah, just less than the first one, the open jar.
Where are the maggots and the flies though? For number
On
2?
the top.
I'm sorry?
On the top?
Yes, on the top, on the netting only, right? So you got all these maggots crawling here. You got these flies, okay? So flies and maggots on the net. Okay, and number 3, no flies, no maggots. Okay, so that's basically the results, right? We are observing the dependent variable in this case. Okay, and in some other experiment, instead of observing, we can be measuring instead, right? So what conclusion was made by Rady? Let's go back to some more vibrant colors here. Meat do not produce flies and maggots. Only flies produce maggots and other flies. Okay, so therefore, the theory of spontaneous generation is rejected. Okay. So he had a hypothesis, right? And it's both testable and potentially wrong. And he did the experiment and showed that it is wrong, right? The theory of spontaneous generation is not true. It's not correct. So then there's another guy, Louis Pasteur. You might have heard of his name, but not realize it. For example, the milk that we drink, or like if you buy honey or something, what term do we use to describe the milk and the honey if they have been sterilized and all the bacteria and germs has been killed? What process do we call that? Anybody know what word I'm referring to?
Pasteurization.
Yeah, exactly, or pasteurization, right? And that's named after this guy, the Louis Pasteur. He basically came up with this pasteurization method. Okay, so what prompted him to perform his experiment? Well, after Rady did his experiment and showed that flies don't come from meat, they only come from other flies. Some people were still not convinced. Okay, they claimed that, well, maybe spontaneous generation occurs on a microscopic level. Okay, maybe it's not true for organisms like flies and fish and stuff like that, but maybe on a microbial level, that is true, right? So what prompted him to perform experiment is that some people still think, and when I say people, I don't mean like the general public, right? Probably it's like people in the scientific community at the time, maybe. So some people still think that spontaneous generation occurs on a microorganism. Occurs for microorganisms, we'll say. Okay, so he's gonna go ahead and try to test that out. Okay. So his hypothesis. So at the time they know about cells already. So if cells arise from non-living things, then they, as in the cell, will appear from sterile broth. Okay, so broth is just like, like a nutrient soup, and sterile means they are heat treated. Okay, so everything is killed off. Okay, so if you killed off everything with heat and then you let it sit there, if spontaneous generation is true, then you will see cells afterwards. That's basically his hypothesis. Okay. So let's see. This is his. By the way, I will post this after class, right? If you miss anything, you can go, find it afterwards. Not to worry. So here we go. In Pasteur's experiment, identify the control group and the experimental group. So he has two groups. He has some flask with the broth in it, okay? And then one of them is open like this, and then the other one has a special swan neck over here. First of all, what is the control group here? Okay, the control group, let me just give you a definition. The control group is the group that has no modification. No modification, okay? The default group. Okay, so if you think about testing the effects of vaccines, the people who do not receive the vaccine, that's the control group. Okay, that's the default with no treatment, no modifications. Okay, so between this flask and this flask, let's call this A and call this B. Which one is the control group? Let me launch a poll just to see if the class is with me. Go ahead. If you think the control group is A, then choose A. Otherwise, choose B. Okay. Anyone else? We have like 19 people who responded. Very good. More people choose A than B, and A is the correct answer. The control group has no modification, right? So he did not attach anything special to the flask. So therefore, this is the control. Okay, the one that has the swan neck, right? This one is going to be the experimental group. This is the one that is going to have the modification, okay, with modification, okay, and the modification in this case is the swan neck tube. That's the modification, okay? So this is the experimental group, the treated group, sometimes also called experimental or treated group, okay? What was the independent variable for this experiment? Independent variable is the one that you are in control of. So what did he control? What did he change? It would be the type of flask opening used. Okay. So does it have a open top like this, or does it have a swan neck? Okay. Next, how did Pasteur ensure the starting condition of all the flasks were identical? Okay, so what he did was all the broth were heated initially to kill off any pre-existing microorganism. Okay. That's how he makes sure that the initial condition were the same. And what were the results? Well, as expected, microorganism were observed in the control group, the one with the open top, but not the experimental group. Okay, and again, that's expected, right? I'll come back to that in a second, because if it's open top, then the airborne microorganism can go in and then they will multiply. The microorganism in the air might come in here, but they cannot climb up the neck, right? Okay, they don't have wings or anything like that, right? So the broth remain sterile. So there you have it. Microorganisms were observed in the control group, but not the experimental group. So he definitively showed that the theory of spontaneous generation is not true. Okay. Does anybody have any question related to either Redi or Pasteur's experiment? Okay, why don't we take a quick yes, Mohammed, go ahead.
Why that the spontaneous generation is not true in that experiment? Because of the meat, the broth is preheated, something like that. For that reason.
I'm really sorry. Your mic is I cannot, because it's in and out. Can you try again, please?
Are you hearing right now?
Yes, go ahead.
Okay, so the conclusion is that theory of a spontaneous generation is not true, because the broth is preheated from the very beginning of the experiment. That's the reason.
Well, because he treated both of them, right?
Mhm.
Okay. So if spontaneous generation is true, And again, spontaneous generation means that the cells would appear on their own, basically, right? So
Right.
if you heat treated everything, you killed off everything, then you would expect cells to appear or microorganisms to appear in the broth, right? If spontaneous generation is correct. But that was not observed, right? He only saw microorganisms in this flask and not this flask, right? And so that proves that the source of the microorganism is actually from the air, right? From the
Oh.
open top, and not that they're appearing out of nowhere on their own. Does that make sense?
Yeah. That's
Okay,
nice.
great.
Okay.
Okay, so let's take a 5-minute break. We will come back at 9:40. And again, for those of you who did not get a copy of the study guide, I just posted a link again on the chat box. You can go get a copy yourself. Okay, so I'll see you in a bit. 5 minutes. Okay, we will come back at 9:41. Hello, let's keep going. So here's the long answer question right here. But before that, let's do a poll. Let me see. Oh, I don't. Oh, there it is. Go ahead. Let's try this question. By the way, I have enabled auto captioning. So if you need captions of what I am talking about, you can turn that on Zoom. Okay, just a couple more seconds. Please choose something. Okay, yeah, most of you got it right. The independent variable is the one that he is in control of, and he is in control of the type of flask that is being used. Okay, he chooses to have the open top or the swan neck, and that makes it the independent variable. The type of broth was not changed in both groups, so that's not a variable. Presence of microorganisms, that would be the dependent variable. That's the one that he's observing. All the flasks were boiled initially, so again, that's not a variable. A variable is something that changes, right? And presence of flies, his experiment does not deal with flies, right? It's the Rady experiment that deals with flies. Okay, here's another example. I think we did this already, so let's move on. Okay, this one is good. Let's see if you can try that. Okay, anyone else? Gonna close it in a couple more seconds. Okay. So remember I said that the test questions or quiz questions, there are the multiple choice, right? Multi-select and application questions, right? So most of the multiple choice question would be something straightforward like these ones, okay? They don't require too much thinking. It's like a matter of fact kind of question, right? And then there are the application questions, which would be something like this, that you have to, really read into it and try to apply what you have learned. So let's see what's going on here. Okay. 1 ml of an experimental drug diluted in a saline solution. Saline just means salt, right? Is injected into 20 pregnant mice to determine possible side effects. Which of the following is a suitable control group? Okay. So in other words, what should we compare this group with? Well, right away, we can reject all the male mice, okay? Because male cannot, they don't get pregnant, right? So if we're trying to figure out the side effects of pregnant mice, we have to use female mice, okay? So non-pregnant, that's not gonna work either. So do we use pregnant mice with 2 ml of the drug or 1 ml of saline? Well, if the drug is diluted in saline and you want to see if the drug is effective, then the benchmark to compare it to would be no drugs at all, right? So we wouldn't test it against something with more drugs in it, right? So the correct answer would be 20 pregnant mice injected with 1 ml of saline.
Mr. Wong,
Go ahead.
it's a 1 milliliter of experimental drug that is diluted in the saline, but it doesn't shows here that what's the saline quantity it is.
Yeah, but the whole thing is 1 ml, right? Like I see what you mean, right? You were like, is it 1 ml of drug diluted in like more saline? But no, this is talking about the entire solution, right?
Oh,
so it's already
it's not clear. That's why I'm thinking that there is no answer.
right.
Because 1 milliliter drug, it is diluted with the saline solution, but what's the saline portion? That is not clear.
Yeah, so this is already within it, okay? Yeah.
Okay.
So that would be the correct answer. Okay, thank you for the comment. Okay, anyone else? Everybody okay? You guys still with me? I'm gonna come back next week. Have I scared you off? Hopefully not. Okay, here we go. Long answer questions. I am going to copy this onto at the bottom, so we have more space for it. Okay, so just gonna copy that. Go all the way to the bottom here. Okay, so you are the lead scientist in studying in a study to determine if taking multivitamin supplement reduces the risk of a cold infection. So part A, write a hypothesis for your experiment. Okay, I'm just gonna do it verbally. This one is pretty straightforward. Again, a hypothesis is a statement, right? A tentative answer to your question. So if the question is, does taking vitamin reduce the risk of a cold? Then your hypothesis would be taking multivitamin supplement reduces the risk of a cold infection. Or it could be taking multivitamin supplement will not reduce the risk of a cold infection. Okay, so you will pick one of those to be your hypothesis. You know what? Let me just write it down here for you. Okay, taking multivitamin supplements reduce, or you can say will not reduce. It's up to you. Reduce the risk of a cold infection. All right, that's that. So part B. What is the independent variable and dependent variable? Okay, please let me know. Anyone? What's the independent variable in this experiment?
Type of multivitamins.
Yeah, either you're taking multivitamins or not, right? Okay, taking or not taking multivitamin supplements. Okay. And what about the dependent variable? What's it gonna be? What are we trying to assess?
The risk of
Infection.
infection.
That's right, the risk of catching a cold, right? So like catching a cold or not. Catching a cold or not. Okay. C, what group should be involved in your experiment? Okay, how many groups should we have for this experiment? How many groups?
Two.
How many, sorry?
Two.
2 groups, okay. Anyone else? Mohammed, I see you unmute. Did you want to chime in? 2 groups? No, okay. Someone else? Prishal? 2 groups? Everybody agree we should have 2 groups? Anyone think we should not have 2 groups? Heinz said 3 groups. That's right. We should have 3 groups. What are the 3 groups?
It's like older people on mid-age like this.
Not quite what I'm guessing at. We're going to get to that in a second. But 3 groups. Okay, we're going to have the group that is, yeah, go ahead.
With multivitamin supplement, without multivitamin supplement, and maybe something else.
Control.
Okay, yeah. What's the word I'm looking for? You guys are getting there, very close. So right now we have 2 groups, right? I hear we said, okay, group 1, take
I think I'm
vitamin,
not taking.
okay? And then we have group number 2, who is no vitamin. Okay, group 1, group 2, and what's the last group? What should we do with them?
I think
Fake pill, fake multivitamin.
placebo.
That's right, placebo. That's right, fake vitamin. Okay, you tell them, hey, this is vitamin, but actually it's just candy, right? Okay, so this here taking vitamin, that is the control group. Okay, sorry, so that's the experimental group, rather. Experimental group or a treated group. Number 2, no vitamin, that's the control group. Right? And number 3, we're going to have the placebo. Okay. So 3 groups. Sometimes the control group is the placebo group. Sometimes you don't make a distinction, but that's what it is. Okay, D, what factors must be kept constant? What factor must be kept constant between various groups? Okay, so these people are taking it, they're not taking it. These are, taking the fig pill, but I have to make sure everything else stays the same, right? So all these other things that must stay the same, we call them standardized. Standardized variable. Okay. The purpose of standardized variable is to ensure all the groups are as close to each other as possible, so that if I observe different outcomes from each of the groups, I know it was just because of the difference in the vitamins and not something else. Okay, so you want to make sure that the effects, if any, that we observe is based on the independent variable and not anything else. Okay, so same conditions, number of people in each group. Yeah, so all these are good suggestions, right? So age, yeah, you don't want all the young people in one group and the older ones in the other. You want them to have the same age group, similar age group. What's another one? Same condition. What do you mean by that, Carl? What do you mean same conditions? Should we lock them up in a facility and then check on them after a week or two? What do you mean? Oh, like, okay, right. So
it looks like in a one specific situation, like in an environmental condition.
okay, so similar health, yeah. Now, when you say environmental conditions,
That means in cold weather or the summer, the cold season or summer, winter season or summer season. I'm talking about.
right. So you want to do that like, within the same time, right? And also you might want to have people who are from similar exposure. Okay, so you don't want to put all the healthcare worker in one group and all the teachers in another group and construction worker in another group. Right? You want to have a mix demographics, right? So that the environment that they're exposed to would be similar, right? So similar health, work conditions, right? Okay, what else? Let me see. Their health, yeah, we said that already. What else? Anyone else have other suggestions? Did I miss anything? Oh, okay, I think I got everything you guys said. I'm looking for a few more, please. Maybe two more. Anyone else? Gender. Give me one more. Body build. Yeah, that would be like, yeah, similar health conditions, right? Environmental conditions. Yeah, we talked about that already. Age and gender, and then usually there's another one that follows. Ethnicity, right? Okay, so people with different ethnicity might have different susceptibility to catching a cold, so you want to have good representation. Does that make sense? So again, these are called standardized variable. It is the things that we try to keep as similar as possible between the different groups. Okay, in the context of Rady's experiment, the standardized variable would be he got to use the same piece of meat. Okay, you got to use the same type of conditions, right? They have to, for example, leave all the jars outside in the same spot, right? He has to make sure that they are left outside for the same amount of time. Okay, so it's easier to control standardized variable when you're not dealing with humans, because humans are all different, right? But we try to standardize it as much as possible. Any questions before we move on? Yeah, time, right? Like you want to start and end the experiment at the same time, right? Like within a month's time or something like that. Okay, and these are all the kind of things that they had to consider, right, when they test the efficacy of various COVID vaccine, for example. What result would support your hypothesis? Well, my hypothesis was that taking vitamin would reduce the risk of catching a cold. So what would support the hypothesis is that people in the experimental group do not catch a cold as easily as the control and the placebo. Okay. So if I see more people getting infected in the control group and the placebo group, then I know the multivitamin is helping. And if I observe anything otherwise, for example, if I see more people getting the cold in the experimental group, or that they get the cold equally in each of the group, then I will refute my hypothesis. Okay, so this is an example of, again, inductive reasoning, right? Okay, we observe different cases. Okay, you know, this person did not really catch a cold, and he was taking the fake pill. This person did not catch it because he's taking the fake pill, did not catch it, and then we come up with all these individual observations, and then we make a broad statement. Oh, so it looks like the multivitamin is helping or is not helping. Okay, we go from specific to general. Okay, so that's that. Let's take a look at this next question. A farmer wants to determine if feeding cows with a high protein diet would increase milk yield. Write a hypothesis for this inquiry. So again, the hypothesis could be feeding cows with high protein diet will increase milk yield, or you can say will not. Okay, it will or it will not. You make a choice. Describe how the experiment should be set up to test this hypothesis. So again, we would have the control group. Control group. Okay. What should we give the control group cows? What should we feed them? What do we feed them, huh? Versus the treated group, the experimental group. Okay. What should we treat? What should we give them? The control group, we should give them more protein or normal protein?
Normal.
Normal protein, that's right. So normal diet. Just the usual stuff that you feed the cow, whatever you feed the cows. And then for the treated group, you would give them
High.
extra protein, right? Extra protein. Okay. Do we need a placebo? Should we
No.
have a, huh?
No,
No, why is that? Why do we not need a placebo?
it's like bull. I don't know, because it's cow. It's an animal. I don't know.
Yeah, you're right. It's animals, right? Like the reason we have a placebo for humans is because humans have psychological effects and bias, right? The cow's not gonna know if you feed them extra proteins or not, right? They're not gonna be like, you know what? I'm gonna hold out on my milk, okay, just to mess with you. They don't know that. They're animals, so there's no need for a placebo. Does that make sense? Okay.
Yes.
I want you guys to try this. Give me 1 second. Just gonna set this up. Give me 1 second. Oops, not working at the moment. Okay, so please click on the link in the chat box. You guys know where the chat button is, right? For the Zoom, click on the chat box and then there is a link there. Click on that link and it will take you to this page. Okay, so I want you to list 3 standardized variables that we should have for this cow experiment that we're talking about. Okay, so remember, we're trying to see if high protein diet would change the milk yield. So we have 2 groups of cows. Cows, we have normal diet, and cows with, you know, a high protein diet. Everything else we should try to standardize. So I want you to go here and type in 3 standardized variables for me. Okay, the link is in the chat box. Please take a moment and try that. Okay, thank you, Justine. I have 3 people who have done it so far. Okay, take your time. I'm going to give you a couple minutes. Okay, maybe just one more minute. Okay, so let's see what people say. Age, the way to read this is the bigger the word, it means the more people. So a lot of people said age. Okay, very good. You want to use cows that are of similar age. Gender is not going to be relevant here, because only female cows can make milk, right? Okay, so like you're not gonna have any boy cows in it, right? Okay, male cows, right? I don't. What do you call a male cow? Like a roan or something? Anyway, you're not gonna have like male cows. Okay? Gender is irrelevant. Food amount, yeah, that's good, right? You want them to have the same weight of food. It's just that one has more proteins than the other. Health condition, good. Health condition again. Health, yeah. Feeding time, that's a good one. Right? You want to feed them the same time, the same amount. Environment, good. Same health issues or lack of health issues. Type of cows, absolutely. Same age. Yeah, these are good. Very good. Locations. Breed. Yep. Good. So you guys are getting this. Excellent. Let's go back to our study guide. Okay, describe the main principles of, it's missing a word here, of experimental design. So in general, when we design an experiment, we want it to be, we want the groups to be randomized. Usually, right? You don't want to target a specific group unless that's the purpose of the experiment, right? Otherwise, you want to randomize. You want to again have mixed age, mixed ethnicity, gender, so on and so forth. You want to randomize it. You want to have large sample size. Okay, you don't want to just test it on 100 people and make a conclusion, right? Okay, like the vaccine, it was tested on thousands and thousands of people before it was made available to general public, right? So you want a large sample size, which gives you confidence in your results, right? You want to have proper groups, right? You must have a control to compare your results to, and it's probably good to do it as a double blind. Okay, so these are some of the main principles of experimental design. That's how experiments are done nowadays. Explain the importance of placebo. We talked about that already, to eliminate psychological effects, to eliminate bias, right? What is the difference between blind experiment and double blind? Okay, so for blind experiment, the subjects, that's the people who are involved in the testing process, the subjects do not know what they are receiving. Okay, so in the context of testing a vaccine, they give you a shot. They don't tell you if this is the actual vaccine or if it is just the a placebo. Okay, so you receive a shot, you don't know what it is. Okay, it's a blind experiment. In a double-blind experiment, both the subject and the experimenter, the researcher will say, do not know. Okay. So I was listening to a podcast that played a clip of when Pfizer, you know, the company, they did their experiment and it was double blind. So like the people who are receiving it, they don't know what they're receiving. And the people who works at Pfizer, who's doing the experiment, they also don't know. And then a third party holds the answer. The third party that has no interest, no financial interest, or they're not invested in the company, right? Third neutral party, they are the one who holds the key to know who received what, right? So the clip was playing the recordings that took place when the Pfizer executive was in this room, and then they were meeting with the third party, and then the third party was announcing the results of the double-blind experiment. It was like, oh, it's 96%, efficacy or something like that. And everybody was like cheering and stuff like that. So that's what double-blind is. Okay, and for obvious reason, right? You want to minimize bias. If I am the company and I've invested all this money in making this vaccine, I naturally want it to work, right? And so, maybe I have conflict of interest there. And if I don't do a double-blind experiment, people would be skeptical of the results that is being released. And even if it's double-blind like it is right now, a lot of people are still skeptical, right? They think, the pharmaceutical companies are conspiring with the government, right, to coerce people into getting the vaccines. But generally speaking, like double-blind experiment is pretty safe, and they are overseen by a governing body, right? So that is that. Okay, here is the application question. The graph below shows the effect of zinc lozenges in reducing cold symptoms duration. Identify additional information that are needed to improve the credibility of the results. Okay, so you see these graphs a lot all the time. Okay, like you see it on like infomercials, for example. You see it in like even advertising billboards that they show the results of how effective their products are. Okay, so if you see a graph like this on the zinc lozenges, okay, see that there's a placebo and then there is the actual lozenges. And you can see people who are taking the lozenges, the number of days it takes for the cold symptoms to go away is less than people who are not taking the lozenges. Okay, so if you see this graph, what additional information would you like to request in order to increase the credibility of this data? Please tell me. What do you think? Do you understand my question?
What was the sample size?
Exactly, right? Sample size, that's a good one. How many people were involved in this study? Okay. Is it just everyone in the company? Is it just, 4 or 5 people, 10 people, 100 people? How big is it? So Victoria says age group. Yeah, so who's involved? Who's involved? Okay, was it young people versus older people, right? What's the nature of the experiment? Okay, what else? Medical history, yeah. So who's involved, right? Background. Their background. Okay, what else? Yeah, time, right? Like when was this done? Okay, was it done during the summer, winter, peak of the cold season? What is it? Couple ones I'm looking for. I'm looking for like 3 specific ones that I would like to hear from the group. How many lozenges were taken? Yeah, okay, yeah, sure. How many do you need to take, right? Do I take a whole pack? Okay, good. Anything else? Yeah, so who's involved, right? The background. Ethnical group. Yeah, very good, Emma. Still, I'm looking for 3 specific ones. So I'll just tell you because we're running out of time. Sorry, yeah. Who's funding the research?
No, I'm talking about that each company will involve with this research because the company reputation is more acceptable by the general people. If I see that, oh, this vaccine is from the Pfizer, then okay, I am mentally satisfied. Or this vaccine comes from the any other countries and it's Asian countries, something like that. No, I'm not taking this one.
Yeah, like right. So like who's funding the research? Who's doing the research, right? Who's manufacturing the lozenges, right? Very good. Methods, yes. So like number of days until cold symptoms were gone, right? How is this assessed? Right? Like, is this just self-reporting? Do you have a doctor assess them? Is there a worksheet that you fill, a checklist that you fill in? How is it done, right? Another experiment is, another thing that we want to look at is, was it double blind? Double blind, right? Okay, so all these things. Okay, just because someone shows you a graph doesn't mean it is scientifically proven. They like to use these, you know, buzzword, right, in products. This product scientifically proven to help you lose weight. Right? Buy it now, and I'll give you another package for free. Anyways, second question here. A new drug called sleep is being tested for its effectiveness in reducing symptoms of jet lag. A double-blind experiment was conducted. The graph below shows the experimental data from the study. Based on the experimental result, what conclusion can be made about the effectiveness of the pill? Okay. So let me launch the poll. Okay. So based on this result, if you think it is effective, please choose A. Not effective, please choose B. Non-conclusive, please choose C. D, not sure. Okay, so everyone should be able to choose something. Okay, let's see. So in the control group, we have 70% of the people who are reporting symptoms of jet lag. Oh, I just found out there's a spelling mistake here, jet lag. Okay, so if you're not taking anything, then 70% of the people reported jet lag. Okay, so placebo, if you give them a placebo, if you give them a sugar pill, they took it, and then only 40% people reported jet lag. And if you give them the pill, right, a little bit under 40% of the people also, you know, there is also a reduction in the number of people who is reporting jet lag. Okay, so both placebo and sleep, the treatment group, have less people experiencing jet lag. Okay, so does it work or does it not work? Okay, the correct answer is it does not work. And how do you know it does not work? Well, because the treatment group performed in the same manner as the placebo group, which means this looks like it doesn't matter if you're taking a candy or if you're taking the actual drug. Both of them is going to help you reduce the symptoms of jet lag compared to control, right? So why would I take the pill when I can just take like a piece of candy, right? So to speak, the sugar pill, right? Because the sugar pill did as well as the drug itself. So based on this result, we can conclude that the pill is not effective. Okay. For the people who chose A as the answer, have I convinced you why The answer is supposed to be B. Yes, go ahead, Natalia. You have a question? Your hand's raised.
It makes sense, but there's still a difference between the placebo and the actual pill.
That's right. So what we can conclude about that is the jet lag symptoms is mostly psychological. Okay. So you can just give people a sugar pill and then tell them, oh, this is going to help you sleep at night. It's going to help you, to get rid of the jet lag. So the person takes it and they think is helping them. And maybe that comforting thought of the pill is helping me actually helps them go to sleep, for example. Okay. So how would the graph look like if the drug is effective? What would be different in the graph if the drug is effective?
It will be above 70%.
What would be above 70%?
The drug graph. Easy sleep.
This one should be like that.
Yep.
Okay. Do people agree
No.
that? Okay, so what should it be, Karina? What do you think?
I think the placebo one should be higher and that the drug one should be low.
That's right. So again, the placebo is a sugar pill, right? So the sugar pill is supposed to do nothing to you. Okay. So if you actually experiencing jet lag, you take the sugar pill, it should do absolutely nothing for you. And it should be as high as or similar to the control group. And if the pill is actually helping you, then it should be significantly lower than both the control group and the placebo group. Okay? So that's how it is. Now, this is explained in more details in the lecture recordings. So please go ahead and watch that if you're a little bit confused. Okay, I know we are a little bit over our time, but I'm just going to take the last couple minutes to fill in the blanks so you have the answers here. Okay, first question. Something is the ability to maintain a stable internal environment despite of environmental changes. That is homeostasis. Okay. Part of discovery science where scientists make generalization from many small observations. Okay, so you're going from little general statement, and then you're going to make it into a general statement. That is induction. This is to make predictions about specific case based on generalization. This is deduction. A tentative, testable explanation, hypothesis. And in order for the hypothesis to be valid, it should be testable and falsifiable. Falsifiable. This variable is measured or observed in an experiment. That would be the dependent variable. And the one that is being changed or manipulated, that is the independent variable. Sham treatment is called a placebo. Something study is one where subjects do not know if they're receiving a treatment or placebo. That would be a blind. And if both the experimenter, the scientist, and the subjects do not know, then we call it a double blind. A correlation is a relationship between two variables. We didn't talk about this during the tutorial, but it was discussed in the lecture recordings. A primary source is a researcher's original report, whereas these things are examples of secondary sources. Okay. That is it for the tutorial. Thank you for coming. Now, just before you go, I would like to remind you that give me 1 second. Just gonna share my screen. Okay. So this is where you can find all the archive videos of the tutorial, right? So last week, if you missed the orientation, you can just click on this and watch it. And today, I'm gonna post it right now after we end. I'm just gonna put the link in the chat box if you don't have it. I will also post a link on eCentennial afterwards. Okay. So just to refresh your memory, if you go to eCentennial after class, not right away, right? Like give me like half an hour or something, right? We have to process the video and all that stuff. So I will have a new news item, and then it will give you all these hyperlinks to click to. Okay. So you can find recordings of today's tutorial here. So you just click on that, and it will take you directly to the page that I was showing you. So obviously, if you watch the whole thing and you don't have questions, that's fine. But sometimes, if you miss a poll or you want to see the poll questions again, or if you just miss certain parts of the notes, you can come here. Also, I will be posting this one in a separate folder, and I'll put a link to it as well. Okay, so that's it. You do not have to come meet live with me next Tuesday. Okay, next Tuesday is asynchronous. You watch lecture 2 recordings on your own, and then we'll meet again next Thursday at 8:30. Okay. If you have any question, please feel free to email me. Otherwise, enjoy the rest of the day, and thanks again for coming. See you later.
Thank you, sir.
Bye bye.
Thank you. Have a good day.
Thank you.
Teacher.
Thank you.
Thank you.